morning, all of you. Good morning, all of you. All of you, can you hear me? I'm audible to everyone. Okay. So today's class will be on vector-borne diseases. What's the vector? So vector is an insect or an agent which is transmitting the infection from one person to another person. So today's class will be on malaria, so which is one of the vector-borne diseases. So malaria, so you all know it's a common term. So it's an endemic disease where we have cases throughout the year. So what are the things you're going to see in this class? So what is the malaria definition, history, problem statement, so parasites, which is transmitting infection, vectors, host environmental factors, what's the mode of transmission, incubation period, clinical features, diagnosis, control and prevention, and recent advances in malaria infection. Definition. So how do you say the patient is having malaria? So malaria is a protozoal disease caused by sporoparasites of the genus Plasmodium. Sporozoa are resistant by intermittent febrile paroxysms, secondary anemia, so patient will be having fever, so which is intermittent type. So we have already seen in a typhoid fever, it will be a stepladder pattern. Here it will be intermittent fever, maybe fever every 2 days, and there will be a gap in between. Another again after another day, we'll be having fever for another 2 days. So there will be an intermittent fever. So what are types of malaria? So malaria, so we have 4 types of malaria. So one is vivax, falciparum, malariae, and oval. So these are all the 4 types of malarial parasites which is transmitting the infection. So malarial parasite requires 2 hosts to complete its life cycle. So it need a man and a mosquito to complete its life cycle. So malarial parasite requires 2 hosts to complete its life cycle. So man, so he is an intermediate host, and female anopheles mosquito, they are the definitive host. Okay, so history. So we say malaria means bad air. So that is bad air means it is contaminated with the parasites. Okay. Fernand Rose is a person who discovered malaria. So problem statement. So world confined to tropics and subtropics. So malaria is common in the tropical and subtropical countries. So more than 90% of the cases are occurring in the sub-Saharan Africa. In India, coming to India, 6.74 million cases to 2.1 million cases are there every year, and we have deaths of nearly 1,000 deaths every year. And API, what's in the API? The API is annual parasite incidence. So annual parasite incidence is 1.62. So we have more number of cases in the northeastern states, and 80% of the population live in the low endemic areas. So types of malaria, so we have tribal malaria, rural malaria, urban malaria, malaria in project areas, and border malaria. So tribal malaria in the hilly areas where tribal people live, rural in the rural areas, urban you all know, and malaria in project areas where the dams and other construction works are going on. So those project areas, and border, so in between the border of 2 different states. So where there will be a construction and stagnation of water, border malaria. So 4 species of malarial parasite are present. So you all know, so Plasmodium vivax, Plasmodium falciparum, Plasmodium malaria and Plasmodium ovale. So bivalves is about 70% India. So very common is bivalves and falciparum 25 to 30%. And mixed infections are about 2 to 8%. And less than 1% we have malaria. So life cycle is asexual life cycle. Asexual cycle. So infected mosquito bites a person and injects the parasite. It goes and bites a person. So when it is biting the person, so they inject the sporocytes which is present in their mouth parts. So after that, the sporocytes enter the blood cycle and it goes to the first phase is called the hepatic phase, where it reaches the liver, and after 1 to 2 weeks it becomes hepatic zygote. So it multiplies in the liver cells and it becomes hepatic zygotes, which is releasing the lymphocytes after that. So from the lymphocytes enter the attaches to the RBCs. So we call it as erythrocytic phase. So which is happening in the liver, we call it as hepatic phase, and which is happening in the RBCs, we call it as lymphocytic phase. Where the lymphocytes attaches the RBCs and passes through stages of trophozoite and zygote. Okay, so these two stages later develops. Gametogeny. Some of the erythrocytic forms do not divide but becomes male and female gametocyte. So here would be the lysis of the RBCs occurs. That's why we say after malaria, the patient may be anemic because lysis of RBCs happening in the malarial infection. Some erythrocytic forms do not divide, so they become male and female gametocyte. They are the sexual forms which are infective for the mosquito. So they are ready in the bloodstream. So when a normal or an mosquito bites a person, it picks up the gametocyte from the bloodstream of the human. So when the gametocytes are ingested by the mosquito, so when it bites a person which is infected, when a normal mosquito bites that person, so the gametocyte is ingested by the mosquitoes. After that, the exflagellation of the male gametocyte occurs, like 4 to 8 thread-like filaments called microgametes develop. So the flagella exflagellation occurs and it develops into the microgametes. The female gametocyte matures and become macrogamete, and fertilization occurs by the process of chemotaxis resulting in the formation of ookinetes. So all these are happening in the mosquito. So this is called the sexual cycle. So the ookinetes now become motile and penetrate the stomach wall of the mosquito and develops into a leucocyst on the outer surface of the stomach. It comes out after the stomach by piercing the stomach wall. Numerous sporocytes are developed in the. So now the sporocytes are liberated in the body cavity of the mosquito. The sporocytes now migrate to salivary glands the mosquito like human infected. So from the stomach wall, the sporocytes become leucocysts, and from the sporocytes liberated in the cavity and they move towards the salivary glands and they become infective and stay there. And when the mosquito again bites a person, normal person, it injects this sporocytes. So again the asexual cycle takes place, and from there again if the mosquito bites the person, so this cycle continues. So asexual and sexual cycle. So now what is about the malarial parasite? So reservoir of infection. Who are all the person who is acting as a source of the infection? Human, who act as a cases and carriers. So he is a case. Period of communicability is 10 to 12 days. So this is how it appears in the blood. So host factors, the age, sex, housing, pregnancy, socio-economic development. Age, mostly all age group are equally affected. And sex is more common in the males when compared to females, as they have less clothing and they tend to move out, they work outside, they sleep outside. So many of the factors are there. So more commonly affected are males when compared to females. socioeconomic development, that's post-socioeconomic development, overcrowding. So all these are another risk factor. So housing conditions where there is places for the mosquitoes to rest, all the no proper sanitation facilities, no proper drainage facilities, and where there is stagnation of water, all these plays as a risk factor for the breeding for the mosquitoes. Population mobility. So population moving from one place to another place. So if the population is susceptible, moving from one place to another place, and if overcrowding is present, they are at risk to develop the infection. Occupation. So people working outside in bushes, in construction sites where there is stagnation of water, in the fields, so where there is breeding of mosquitoes are present. So it's very common. So those occupations are common for the people to get infected. Human habitats, tribal areas, rural areas, slum. So these are all the habitats where they are easily prone for the infection. So we have natural immunity, and so to some extent that will protect us for the next infection. So environmental factors. So season. So more common during the rainy season where there is stagnation of water. So more common for first 6 months. And there is heavy rainfall or stagnation of water is present throughout the year. So season is very common. And temperature. So normal temperature, humidity, and more rainfall. So some more rainfall may be unfavorable for the mosquitoes as they are so moderate to light to moderate heavy rainfall is okay, but still if there are very heavy rainfall, the breeding places may be washed out. So that is one of the what is there. Okay, so coming to the environmental factors, so season. So it's a seasonal disease where it is very common from July to November. So July to November where there is heavy rainfall. And temperature, so it's common between 20 to 30 degrees centigrade. So this temperature is very favorable for the mosquitoes to breed. Coming to the humidity, 50% humidity is necessary for the mosquitoes for them to live and to have a normal lifespan. Humidity is necessary for these mosquitoes. Rainfall, so in general, it provides breeding place for the mosquitoes. However, heavy rainfall as already I told you, it has adverse effects of flushing out all of the breeding places. In altitude, so anopheles are not found in high altitudes, that's from 2000 to 2500 meters. So it is unfavorable for those mosquitoes to breed and live in those conditions. So what's in the man-made malaria? Burrowing pits, garden pools, irrigation channels, projects. So where man made all this construction and made water to stagnate and store in those places, mosquitoes can breed. So that is called as man-made malaria. And coming to the vector, so vector you all know it's a female Anopheline mosquito. So that is the vector which is causing the infection. And life history of this vector. So, and what is the density? So, density should be more for the vectors transmit the infection. So, adequate density near the human habitation. So, then only they can transmit the infection. And what about the lifespan? So it's a key factor for transmission of malaria. So as long as they live, they keep transmitting the infection. If they have a very short lifespan, so they don't transmit much. So at least they need 10 to 12 days of lifespan to transmit the infection. The choice of host, that's feeding habits. Choice of host, some mosquitoes prefer human blood when compared to the animal blood. So when they want for the human blood, so there is more biting of the mosquitoes. That depends upon the choice of the feeding habits of the mosquito. Resting habits. So you all know resting habits after a blood meal, some mosquitoes rest indoors on the walls for quite some time. So they rest indoors and some rest outdoors. So the behavior pattern is known as endophilia. So those what about the breeding habits? So breeding habits of mosquitoes has to do moving water, stagnated water, and they also breed artificially stored water. So different mosquitoes breed in different places. So anti measures are used in all these different types of water sources that are time of biting. The majority of Indian mosquitoes bite at night excepting for the Aedes mosquitoes. Aedes mosquito transmits what? Aedes mosquito transmits dengue and chikungunya. So these mosquitoes bite during daytime. Whereas all the mosquitoes are night biters. They bite night time. The vectoral capacity. The term vectoral capacity refers to the combined effective density of the population and susceptibility to the infection. Resistant insecticides. Knowledge. So some of the this depends on the choices of insecticides which we use. So some insect vector are resistant to the insecticide and alternative insecticide usage so that to prevent resistance of this insecticides. So next one will be mode of transmission. So how it is transmitted. So vector transmission, direct transmission, congenital malaria. So we have vector transmission. So malaria is transmitted by a bite of a certain species of infected female, so which is an anopheline mosquito, and it's a single infected vector during her lifetime can infect several persons. So until the sporozoites are present in the salivary glands, the infected mosquito can transmit the infection. Direct transmission, malaria can be induced accidentally by hypodermic, intramuscular, intravenous injection of blood or plasma. So blood transfusion, drug addicts. So this is through the direct transmission. And congenital malaria. So congenital infection of the newborn from the infected mother. So we can have this congenital malaria. So incubation period is not less than 10 days. Okay. So incubation for different types of parasites. So falciparum 12 days, vivax 14 days, malaria 28 days, mobile 17 days. So these are clinical features how the patient will present with. The patient will present with fever, anemia, jaundice, nausea, vomiting, headache, and other complications can occur. So clinical features, so in first stage is called as cold stage, where the onset is with lassitude, headache, nausea and chills. And temperature rises by 39 to 41 degree, 1.5 hours. So it's cold stage and then hot stage. Hot and dry headache and rapid pulse present and lasts for 6 hours. Later on, they'll have sweating stage. Fever comes down with profuse sweating and lasts for 2 to 4 hours. So these are the classical clinical features of malaria. Called as cold stage, hot stage, and sweating stage. Now the areas of the hospitals are laid. Pupa, hospital, the mouth parts, how to differentiate between male and female mouth parts, resting habits, and what are the sources of breeding places in hospitals. Small stagnation of water also will cause it's the place for breeding for the mosquitoes to breed. Mostly the agricultural lands where paddy and other things are grown, where there is stagnation of water throughout the field, where it happens. So the that's why we ask them to go for intermittent irrigation. So agricultural farmers as they will have stored the water for 1 week. Later on, they will let it dry for another 2 days, and after that, again they can store the water. So this they can stop the breeding of the mosquitoes. This consists of dams, parks, and other things where there is storage of water, artificial fountains. All these places are the breeding places for the mosquitoes. Common the urban malaria. So where there is presence of plants and plants are not closed properly, that will act as a source for the laboratory diagnosis. How we are going to diagnose? The blood smear, which is a thick and thin blood smear. The clinical diagnosis, an antigen protein test, and PCR test, and serological test. So which we can diagnose the infection. So we can think here, we can find the parasite and the role of the parasite. So what is the parasite which causes the infection, and what's the density or the role of the parasite plasmodium plus. This is how we make the slide. Explain. Prevention and control. First, we measure the burden of the malaria. So, in a pre-eradication era, before the eradication happening, so we will consider million screen rate because that is the environment of screen after the malaria infection. So, average enlarged screen rate, parasite index, and parasite rate and proportional case rate. So, these are all the parameters that we measure for the burden of the malaria cases in the country. The eradication era will go for annual parasite incidents. How many of them are infected annually? And blood examination rate. How many of the blood cells are examined in a year? And annual falciparum incidents. How many of them have falciparum malaria? And slide positive rate. How many of them have found slide positivity on the examination? And slide falciparum rate. How many of them have shown positive for the slide falciparum? Vector indices, blood index, parasite rate, mosquito density, biting rate, these are the vector density indices. So approaches and strategies of malaria control. So management of malarial cases. How we are going to manage the malarial cases and prevention of malaria transmission in the community. How we are going to break the chain of transmission. So how we are going to manage the malaria cases here. So disease control strategies and we can find case detection. So case detection. How you will find the case detection? So 9 byte survey, 9 byte survey. So active and passive surveillance. So by active surveillance, you go house to house and take the 9 byte survey stick and thin square and identify for the malarial parasite. So passive surveillance, hospital, reporting fever and all the signs and symptoms of malaria. You will take the blood smear and do the examination to find the cases. So that's how you will detect the cases. And treatment, we go for the high risk case, low risk areas, and severe and complicated malaria. According to that, we go for the treatment. Mass drug administration and chemoprophylaxis. The stratification of the problems, areas with APA less than 2 and APA more than 2. So animal parasite incidence less than 2, so that's a different mode of control measures, and API more than 2, different mode of control measures. Vector control strategies, anti-larval measures, anti-adult measures, and protection against mosquito bites. So anti-larval measures, so we have all this biological, chemical and everything, and anti-adult measures also, and protection against mosquito bites, individual protection. So anti-larval measures, environmental conditions. So that we have this chemical control where we have mineral oil, paraffin and synthetic insecticides which is sprayed in the where there are water areas where breeding for the mosquitoes are present. So breeding areas we spray all this oil, so mineral oil, insecticides and all these things are sprayed so that you can control the larva which is present in the breeding place. And biological control by introducing all these larvae fishes in the ponds and wherever necessary, and bacillus, thuringians and fungi, ferns, everything can be introduced so that they will kill the larva. So this is coming under anti-larval measures of environmental control, so chemical control and biological control. So deweeding, desilting, don't allow water to stagnate. These are the various fishes which is introduced. Next one is anti-adult measures. So residual sprays. We have this residual sprays which is sprayed inside the houses where the adult mosquitoes flies. So when you spray from outside, they come inside indoors and rest indoors. So you go for the residual sprays where DDT, malathion and fentanyl is used for the residual sprays. And spray sprays is pyrethrum by ultra low volume method. So you would have seen this fogging method where they go every street and they use this fogging to kill all the adult mosquitoes which is present outside. Individual protection. So individual protection by the proper clothing. And genetic control to reduce the multiplication of mosquitoes laying eggs. This is ultra low volume fogging. This is spraying. This is individual protection. So how we are going to protection against mosquito bite, site selection, mosquito net, screening and repellents. So you have all these methods. You can screen your houses, windows, doors, use repellents, mosquito nets. All these are coming under individual protection. We have national program which is related to this, a national malaria eradication program. That's vector-borne disease control program. So under this, all the vectors are controlled under the single heading, which is called as integrated vector control program. So everything comes together, like chemical, biological, environmental, person protection, and health education. All these measures are together we call as integrated vector control program, which is a very important shortcut. You can be asked in your exam. What are the recent advances? So we have this malaria vaccine, synthetic cocktail vaccine SPF 66 and candidate vaccine EFS 25. So these are all the recent advances where malaria vaccine are addressing. These are the references. Okay. So now this is end of the class. So you can go through the book after the class so that you will be able to understand. So I want all of you to prepare some short notes and write in this topic: treatment, API, active and passive surveillance, national malaria eradication program. So all these are the important topics, integrated vector control measures, so which has to be learned. So these 4 to 5 topics, you please take down and take the notes and submit. So my other vector control program will be read in the other classes.